Hello and welcome to The Constant, where the only constant is change, and we go for a swim in the deep blue of business technology and culture. I'm German Dziebel. From new technological innovations such as artificial intelligence and new business models cropping up in Silicon Valley, New York, and globally, to disruptive thinking in science and creative developments in culture, The Constant podcast analyzes the world we live in from a social, scientific, evolutionary, and philosophical perspectives. In the US, we're used to the out of the box technological and business thinking coming to us from the West Coast, California, and the Silicon Valley. We use iPhones, take an Uber from the airport, stay at an Airbnb, shop on Amazon, and envy people who drive a Tesla. All these companies started with very humble beginnings. The famed Hewlett Packard originated in a private garage in Palo Alto, California in 1939, inaugurating an era of technological disruptions and startups. History shows that successful innovations first meet with resistance, neglect, and even hate, and they have to overcome institutional hurdles and cultural stereotypes before they break out and become big. And many innovations begin with breakthrough thinking in science and culture, and then they trickle down into technology and other practical applications. Our today's theme is maximum genetic diversity theory and an out of Asia model of modern human origins and dispersals. The maximum genetic diversity theory challenges two dominant and interconnected lines of thinking in today's evolutionary genetics: the neutral theory and the associated molecular clock, as it is called, and the single origin of modern humans and their dispersals from Africa between 100,000 and 50,000 years before present. If proven correct, these theories will have impact not only on our general understanding of who we are as a species, how species evolve, how complex systems operate, but also on such practical applications as medical genetics. Our guest today is Dr. Shi Huang, who over the past 30 years has been developing this radical alternative to neutral theory and the recent out of Africa model of human origins. Shi Huang received his PhD in biochemistry from the University of California, Davis. He was a faculty member of the Sanford Burnham Medical Research Institute in San Diego for over 15 years. And since 2009, he has been a professor of genetics at the State Key Laboratory of Medical Genetics at Central South University in Changsha, China. At the end of last year, Dr. Huang's theories were popularized in the science section of the South China Morning Post, and today he is here with us at the Constant Podcast. The world is closely watching competition between the US and China in artificial intelligence, with the DeepSeek challenging ChatGPT for world dominance. Now we may have a similar competition brewing in the scientific field of human genetics. Welcome, Dr. Huang.
Hello, German. Thank you for the opportunity to talk to you. It's great to finally meet you and talk to you in person.
Likewise. Dr. Huang, at the Constant Podcast, we like to look at root causes. Could you describe the origin of your theories? What is your personal story? How did you arrive at them? What gave them the original impetus?
Yes, I had a long interest in evolution. As an undergrad, I majored in genetics at Fudan University in Shanghai. The department was led by Professor C.C. Tan, who was a PhD student of Dobzhansky and Thomas Morgan of Caltech in the 1940s. Dobzhansky was one of the founders of the modern synthetic theory of evolution, merging Darwin's theory with genetics. Therefore, the genetics program at Fudan has a very strong focus on evolution. We learned Darwin's theory of natural selection, Kimura's neutral theory, as well as the molecular clock hypothesis. But at the time, I only had a vague or superficial understanding of these theories and considered myself only a casual believer. I then came to the US for a PhD in biochemistry and molecular biology right after graduating from Fudan. I then had a career in cancer molecular biology for 20 years. Right at the time I started my lab in 1992, we had cloned a novel cancer gene, PRDM2. We have since then been constantly searching the gene bank database for homologous genes of PRDM2 from different species. Around 2005, we first used the human PRDM2 protein sequence as a query to search for homologous proteins in the gene bank database, which is a routine practice. We obtained the expected alignment identity results, showing that humans are closer to frogs than to fish. Next, we conducted an unconventional experiment by using the fish RIS1 protein as query, fully expecting, like most researchers would, that fish would be closer to frogs than to humans. To our big surprise, we observed the genetic equidistance phenomenon, the GEP, with fish being approximately equally different from both frogs and humans. For a short moment, I was very excited as I thought we had made a big discovery. But we soon realized that this was an old phenomenon which has inspired a theory to explain it. That is the molecular clock hypothesis and the neutral theory. But the tricky, unusual thing about this is that the theory behind the phenomenon, behind the equidistance phenomenon, is not presented as an explanation for the equidistance phenomenon. Rather, the molecular clock was presented as a factual phenomenon rather than just a hypothesis. Consequently, almost no one today knows the genetic equidistance phenomenon. But we soon realized that the molecular clock explanation is merely a tautology. After 3 years of in-depth research, we came up with a new theory, the maximum genetic diversity theory, to explain the genetic equidistance phenomenon. We also later identified a new characteristic of the phenomenon known as the overlap feature, which has not yet been recognized or explained by existing theories.
Interesting. Stories of how theories, come about, are truly fascinating. And it would be great, to hear a little bit more about. So, what is in a nutshell the difference, between the maximum genetic diversity theory and the traditional thinking? We've all been taught neo-Darwinism, the synthesis, neutral theory, the Hardy-Weinberg equilibrium as unshakable foundations of modern genetics. The molecular clock has been a contentious issue with multiple algorithms surfacing over the years. And I remember at Stanford in the early 2000s, Lev Zhivatovsky's algorithm was very popular, but then it faded away, did not withstand a closer scrutiny. And now geneticists are using a different algorithm for calculating the divergence of lineages. So there is some flux, with the molecular clock. There's always some uncertainty. But the premise, that genetic diversity accumulates with the passing of time at a steady pace has been a received wisdom. So what are your fundamental disagreements with this paradigm?
Well, yeah, the central issue is whether the observed genetic distance, which is measured by sequence dissimilarity, whether it has an upper limit. The molecular clock hypothesis tacitly assumes that no such limit exists. However, evidence suggests that a limit does exist and that, in most cases, current genetic distances are already at this upper bound. This is the core claim of the maximum genetic diversity theory, a point explicitly emphasized by the theory's name.
Yes, that's a true differentiator for sure, because definitely the neutral theory, does not assume or ever thought about or even disprove the possibility of an upper bound, because it is assumed, to be infinite and ongoing, and the creation of diversity is supposed to be infinite. But how can we empirically differentiate between the two theories? What are the facts at hand that show that there is this upper bound to the accumulation of genetic diversity? And why has it been overlooked? Why is there this tendency, to think, of genetic diversity as accumulating without any limit?
Right. There are actually three different tests we have done in recent years to differentiate between our explanation and the neutral theory or molecular clock explanation. The number one is called overlap feature. It means independent mutations hitting the same site. So the mutation overlaps at the same site among different lineages, which is a rare occurrence if the neutral theory is correct, because it implies an infinite site model that repeated mutations at the same site just do not occur under that model. The second, I'll add that overlap means in reality, what you observe is that there is an abundance of overlapped mutations, which is not expected under the neutral theory, but is predicted by the maximum genetic diversity theory. The second evidence is called the genetic non-equidistance despite equidistance in time. According to the molecular clock, if you have evolved the same amount of time, you should have similar level of genetic distance. So the genetic distance is linearly scaled with time. And however, what can be found is in many species, for two sister species within a clade, their distance to an outgroup reference species is related to their complexity level. Not so they're not equally distant to the outgroup if their complexity, if the sister groups differ in complexity, even though they are separated from the outgroup by the same amount of time, yet they do not show the similar genetic distance. This is unexpected from the molecular clock, but is predicted by our theory. Number three is one can show that ancient genomes is more distant to a reference outgroup than its extant sister species. This is not expected from the molecular clock. So there are many evidence supporting our theory, and all these tests are done by us, not by the other party, our competitor, because they never entertained the thought of an upper limit on genetic distance.
Yes, that's exactly what I wanted to point out because I've never heard about any of that. I mean, I only hear it from you and from the studies, that you've published. But definitely it has never been taught, discussed in papers. The upper limit, the dependence, of genetic diversity upon the complexity of an organism. Those are a very, very new and unexpected angles. Very often in science, we need to be aware of what we don't know, and there is a tendency in science to kind of paint a picture that's a complete picture, and as if there is nothing else to know. There are no other factors, there are no other angles, there are no other phenomena that may impinge on the processes at hand. And what you're bringing up, even as a hypothesis, even although it's a hypothesis based on empirical evidence, is that there is a such thing as complexity dependence. There is a such thing as upper limit. So those, need to be addressed within the broader collective of scholars to see what has been missed. And it's not the only area we're encountering this problem, because in general, innovative thinking out of the box, reaches beyond the current horizon of what people think about. It's not that it continues a line of thinking and just kind of evolves on it. It actually leapsfrog the current thinking and introduces new dimensions for people to think about. Even if we look at business, where there is a theory, of blue ocean thinking, where a company establishes itself in a completely new category, defines that category. All the major disruptors in modern business started as from the blue ocean thinking. And here's what I'm sensing here in your theory. So you're establishing new parameters that need to be interpreted, if we want to get at the correct interpretation of species divergence and the time, that involved, that divergence. But how would you characterize the relationship between the two theories or the two paradigms? Are they totally diametrically opposed to each other, or does the maximum genetic diversity theory include reasonable and empirically supported aspect of the neutral theory? If so, how should we think about them? Is the maximum genetic diversity theory an alternative to the neutral theory, a complement to it, or it's evolution? What are your thoughts?
Yes, we are not totally overthrowing the neutral theory. Neutral theory has great value as a null hypothesis for explaining, for testing natural selection of variants. So for that, it will be always with us. However, its claim as an explanatory theory of nature is totally wrong. So it's not an explanatory theory. And genetic diversity pattern in reality cannot be explained by neutral theory. And so to explain the genetic diversity pattern, we think the maximum genetic diversity theory explains the nature better. Simply for the reason that genetic diversity pattern we observe today are mostly at maximum optimum level of is at equilibrium. So it's not scaling with time. And so in this regard, MGD, maximum genetic diversity theory, is claiming very different things. And also, big difference is that the neutral theory treats most of the genomic sequences as neutral or junk DNA, whereas our theory claims that most of the DNA are functional or under selection. And also, one point we also grant the neutral theory to be true is that for a linear stage evolution where the maximum level has not been reached, for truly neutral variants and for early stage of divergence, the neutral theory should still hold.
Okay, well, that's interesting. That reminds me of the theory of punctuated equilibrium, in a sense, and it may be a distant comparison, but what I'm hearing you're saying is that there is, in a sense, a combination of stretches of evolution governed by neutral theory and stretches of evolution governed by the maximum genetic diversity theory. So there is a linear progression according to the neutral theory predictions, then it plateaus and in a sense stops accumulating diversity at the same rate as before. Is that an accurate statement? It's interesting what you said. It reminded me a little bit
Okay,
of the
great.
punctuated equilibrium theory that thought that evolution occurs, in different modes in a sense. So there is one mode, when it evolves fast, then it stabilizes, slows down, so that there is an inherent kind of change of pace within evolution. And it's a rather widely discussed and widely adopted theory, and it has gone beyond biology into other evolutionary fields. Because what you said made me think that the neutral theory describes the original evolution of a species pretty appropriately, but then there is a plateau moment when maximum genetic diversity is achieved and it's kind of stabilizes. So what happens after that? And do you agree in with the comparison of maximum genetic diversity theory and the punctuated equilibrium thinking?
Yes, I think there are similarities between the two ideas. And so in our theory, we define microevolution and macroevolution differently. So macroevolution is mostly genetic changes, accumulation of variants over time until it reaches equilibrium level and stabilizes. And it accounts for minor adaptive changes within a species or among species, like changes from one species of a monkey to another species. However, for macroevolution, we refer mostly to advances in complexity, in organismal complexity, from chimpanzees to humans will represent macroevolution with a change in complexity level. What change implies at the DNA level has been for a long time unknown. We have this novel idea that with increase in complexity, there must be a corresponding decrease in genetic diversity a species can tolerate. And this change in complexity is mostly due to epigenetic programming changes. And this epigenetic change generally is fast and sudden. And so in this sense, it's like the punctuated equilibrium
Yeah, that's
hypothesis,
interesting.
right?
So let's
Yeah.
look a little bit deeper into the phylogenetics. How does the maximum genetic diversity theory apply to phylogenetics? And maybe we can think about two different examples. Let's start with interspecific comparisons between species. What can MGD tell us about the order of primates, including platyrrhines in South America, and the hominid family? So we're looking at the monkeys and then the hominids. What can you tell us about the role MGD plays in rethinking some of the established patterns of clustering within those two domains?
Right. Traditionally, according to the molecular clock hypothesis, uh, and as well as the neutral theory, most of the DNA sequences can be used for phylogenetic inferences. And because they treat most of the DNA as neutral. And for phylogenetic inferences, you need a neutral DNA to do the analysis to be informative, because functional DNA are under natural selection and they may not reflect the time of divergence. But the flaw there is most of the DNA are in fact not neutral. And there's only a very small fraction of DNA that are still neutral. They are slowly evolving and they are neutral. Therefore, the difference between our approach from the field is that we need to carefully select the truly neutral sequences to do the phylogenetic inferences. And therefore, we can obtain very different results based on whether we select the neutral sequence or the non-neutral sequence. Therefore, we got very different phylogenetic tree from the method used by the field. So in a way, they used the functional sequences. We actually use the neutral sequences. So for hominids, traditionally humans are grouped with chimpanzees, and we consider chimpanzee to be our closest relative. But that is based on whole genomic sequence similarity. It's true that chimpanzees is closest to humans in genomic DNA, but most of this genome DNA are functional. So the similarity there between us and chimpanzee may reflect uh, functional similarity or phenotypic similarity rather than phylogenetic closeness. And to use slow neutral sequences to reanalyze the relationship, we found that chimpanzees are actually closer to orangutans than humans are. So we based on that results, we grouped chimpanzees, gorillas, and orangutans as one clade to the exclusion of humans. So this is traditionally called the Pongid clade, and it's more consistent with trait anatomy-based phylogeny. So there's a unification of genetics and morphology there. So for the primate, another result we obtained that's different from the mainstream result is that tarsiers actually group with prosimians rather than with the higher primates like monkeys. Again, using the same slow clock method.
That's interesting. It gives a sense, how the two, what is the difference in practical outcomes between the two theories. So there is an alternative, and I'm sure, kind of the separateness of humans from the chimps is probably something that we intuitively can agree on.
Exactly. Yeah. So whenever you have a clade of species, like a monkey clade or the ape clade, you will find that some species among those would be closer to the outgroup human. And that's just due to convergent evolution. So the difference is minor, but it's still observable. So for the Pongid clade, you find chimpanzee to be closer to human. So that's just a reflection of adaptive evolution. And for the monkey clade or the New World monkey clade, you see the same thing. Like the owl monkey is closer to human than the marmoset. And you can have examples like this in other clades as well. Like octopus is closer to human
but
than
that's not
clam.
phylogenetic, obviously that's convergent, right? That's the yeah.
Right,
What's interesting to
exactly,
me is that the
exactly.
genetic
And
distances don't
yeah.
necessarily represent the full reality of species divergence, and correct me if I'm wrong, as populations.
Well, genetic similarity could mean two things. One is trait similarity, right? Because phenotypes are determined by genetics, by DNA. And so if you see two things very similar in in DNA, you can infer that they would be similar in traits and phenotypes. And so DNA similarity reflects also, of course, phylogenetic distance. However, you have to use informative DNA for that inference. You have to separate the DNA that reflects functional similarity and the DNA that reflects closer phylogenetic distance.
What it made me think is that in her book, The Wrong Ape, K. Martin argued that the chimpanzees do not provide an optimal morphological and social model for the earliest human ancestor. So, for instance, male dominance that's pervasive among the great apes, say for bonobos, contrasts with cooperative breeding and paternal care that's found across human species, across human societies. And obviously, cooperative breeding, paternal care is found in other species as well, outside of the primate group, among elephants. But within the primate domain, it's interesting that the chimpanzees, do not exhibit the same social organization in its fundamental forms, as the human societies. Most curiously, the closest examples of human social behavior, including, as some primatologists argued, language, comes from New World monkeys, primarily Cebidae and Callitrichidae. Evolutionary anthropologists have strongly argued that it's not so much solitary stoneworking. For many years, that was the kind of the paradigm of thinking how we evolved cognitively. But it's intense social interactions and the recognition of others that involved in cooperative breeding and paternal care that have shaped our human brain. What it sounds like is that convergence is a more powerful force in evolution than we have thought, and that some traits, including genetic, that we tend to ascribe to common descent with chimps, are in fact homoplastic in nature. So we need to kind of, to your point, clearly separate, the kind of genetic information that will express itself in the phenotype, and both will carry descent information, and then others where there is a discrepancy and it's pretty much kind of homoplastic and irrelevant for the assessment of descent.
Yes, homoplastic evolution or convergent evolution is much more common than originally thought. Because neutral theory interprets evolution as non homoplastic. So that means repeated mutations do not occur. So there you have excluded the possibility of convergence. Convergent evolution would be common, especially for fast evolving functional sequences, because they play adaptive roles. The turnover of alleles is very fast, much faster than previously expected. This has been recently shown by several papers about fast adaptive changes.
That's interesting because
yeah.
So we now have two major, not one kind of common descent by default, with some convergence sometimes happening, but two major forces in evolution, obviously descent and convergence at the same time. So we need to differentiate, between the two and keep track of both, and have enough robust empirical data to assess, whether it's common descent or convergence. Because currently, yes, convergence is being put, on the back burner. Earlier, you talked about the infinite site theory.
Right. Whenever they see DNA similarity, yes, whenever the field sees similarity, the automatically assumes that's all due to shared common ancestry rather than adaptive convergent evolution.
Yes.
So that's incorrect.
Yes. Okay. That's very clear and important. And now, Dr. Huang, I think let's move over to one of the most radical applications of the maximum genetic diversity theory to date, by which I mean your reconsideration of modern human origins and dispersals. And this topic obviously commands, the greatest public attention, and it impacts a lot of different fields, around population genetics. As we all know, that used to be, that archaeology and paleontology were the central disciplines defining through the fossils the major sources and expansions of modern humans and the evolution of modern humans, from a primate ancestor and its dispersal around the globe. Since the discovery of the non-recombining nature of the mitochondrial DNA and the typing of different human populations, genetics has progressively become the central, most influential field in the overall modern human origins and dispersals paradigm, with paleontology and human biology, physical anthropology taking a back seat. And I see it by talking to many professionals, working in the fields of the evolution of language and the fields of comparative mythology, comparative culture. So all the eyes now are on population genetics. And it has come up, kind of a party line, of an out of Africa theory. Could you elaborate on this? And how does the maximum genetic diversity theory change that fundamental perspective that we have on modern human origins?
Right. People like the genetic model simply because they consider genetics to be more like hard science, whereas fossil interpretation may be more subjective. That's one reason, the big reason for the out of Africa theory to be triumphant over the alternative fossil-based multi-regional hypothesis. And the out of Africa theory, therefore, is primarily supported by genetic evidence rather than by fossil evidence, right? And the strongest genetic evidence for supporting this the out of Africa model is the high genetic diversity of Africans. And this is based on the neutral theory and the molecular clock, because genetic diversity scales with evolutionary time. So high genetic diversity means the lineage has evolved very long time. So this is the theoretical basis for inferring from high genetic diversity to the origin in Africa. However, the flaw here is that the genetic diversity we do see today in Africans or as well as in other population groups, they are all at the upper bound level, the maximum optimum level. So once they are at the maximum level, they are no longer related to time of evolution or a mutation rate. So one cannot infer from the maximum level of genetic diversity, infer the time of evolution. And therefore, just because Africans have higher genetic diversity does not mean necessarily that they have evolved they have evolved longer than other human groups. So, so if the neutral theory is flawed, then the out of Africa model automatically collapses. And we have used different DNA sequences to study the problem human origin. We use the truly neutral, slow evolving variants to do this. And we can see that human population groups have separated for a long time, for 2 million years, based on these informative markers. And so that's kind of consistent with the initial migration out of Africa for the Homo erectus. So in this sense, these are based on the autosomal markers. So in this sense, it's very consistent with the multi-regional hypothesis, claiming that humans have evolved independently for 2 million years in different areas of the globe. So that's based on autosomal data, and then it turns out the uniparental DNA has turned over several times during this 2 million year period. So modern human mitochondrial DNA and Y chromosome are more recent in origin and have a single place in origin. We have placed it in East Asia rather than in Africa. So we can redraw the phylogenetic tree of the uniparental DNA with the root now in East Asia rather than in Africa.
Interesting. So that reminds me of the original paper by Johnson et al., 1983, on mitochondrial DNA morphs, as they were called at that time. So the study was done at Stanford under Luca Cavalli-Sforza, and for a good decade or two, it was the most cited study of modern human populations. And what that paper brought up at that time are two possibilities to interpret the mitochondrial variation. One, based on the central type, the most widely distributed sequence of genetic changes, before they were even called the haplotypes, and then the type that's most divergent. So if the most divergent type was used, then the tree was rooted in Africa, and ultimately that theory prevailed. But the Johnson et al. 1983 paper also contained the central type possibility, and if we root our tree in the central type, the most common type in modern human populations, so then the root lies outside of Africa, closer, to Asia, Eurasia, America, in that area. So it is fascinating, that your thinking was prefigured in that original paper, but it just never has taken hold. And the academia started focusing exclusively on the out of Africa model. And I'm kind of curious, what are your thoughts on why that original out of Asia or out of Eurasia option was thrown under the carpet?
Yes, these two different interpretations, one based on the central type, the other based on high genetic diversity, are contradictory to each other because they are not unified. They do not point to the same conclusion. And so exactly one points to East Asia or America, right? And the other points to Africa. And so the 1983 paper was actually put forward both models. They explicitly said that if they assume the molecular clock and neutral theory, they can get the African model, African origin. But if they consider the molecular clock to be wrong or invalid, then the central type prevails. So the 1987 paper just totally disregard the central type without explaining why the central type is not a
Yes.
reasonable hypothesis. So with no explanation, they believed in the molecular clock interpretation of genetic diversity. So that's how the out of Africa model become popular. Exactly how you get away with the field with the everybody, even though it's controversial. Molecular clock has always been controversial, and the neutral theory is also a controversial theory. And yet, all these controversies are not discussed, and
Yes. Yes.
so no one really knows what happened. But
Well, here is a possibility, but before I go into it, so my question for you is that, so if Africa has more genetic diversity and Asia has less genetic diversity, does it mean that by that token, the Asian population is older than the African population because it achieved kind of a new level of complexity that slowed down its accumulation of mutations, whereas Africa is still in that initial stage of more linear accumulation of diversity that hasn't stabilized yet? Is that how we should interpret the maximum genetic diversity?
no. That's we should consider both, whether it's high genetic diversity or low genetic diversity. They are both at the upper bound level. It just means their upper bound are different.
Okay, okay, so the upper bounds
So
can be different from population to population, right? Okay.
exactly.
I came up with the hypothesis that the reason why the central type that pointed to East Asia as a potential source of modern humans and Johnson et al. 1983 was not taken into consideration seriously, and there were no papers really diligently comparing the two options, the out of Africa from the most divergent type and out of Asia from the most common type,
Mhm.
was that if taken to its extreme, the central type logic may have pointed to America as being the least derived human population that had frequency of the central type at the highest. Because originally, so in Johnson et al. Paper, they could not really sequence, one restriction enzyme, H2, for the Warao Indians, which was their proxy for the American Indians in total. So then they managed, to come up, with another sample from Southwest American Indians, and that showed, that at H2 the morph, was the ancestral morph at 92% frequency in those American Indians. So then, and that was a Wallace et al. Paper from 1985. And then the French researchers, Escoffier and Langany, when they pulled together Johnson et al. And Wallace et al. 1985, they built a combined table where American Indians were found to have in that central type at the highest frequencies. If we interpreted it at that time, and we collectively, literally, that would have pointed to America as being the center, for human dispersals. But at that time, and currently considered to be a sheer impossibility for a completely different set of reasons, because it doesn't have early hominids. Asia has plenty of those. Africa has hominid fossils. America does not have it. Plus, America doesn't have a good, well understood archaeological record that stretches beyond 15, 20,000 years. So that, could not have been put on the table. And so my hypothesis is that it kind of scared people away from thinking about Asia and the central type as the root of the human phylogenetic tree for mitochondria. So what are your thoughts on that? How would you answer people saying that the Johnson et al. Paper and the subsequent papers actually pointed to America having the most ancestral type at higher frequencies than Asians? What are your thoughts on that?
Yeah, that could be a possibility for explaining why the central type was not taken seriously. But to explain the high frequency of type 1 in America, maybe one of the reasons is they have sampled not enough people. So the limited sampling may have biased the result. And also later studies with more modern information and more defined mitochondrial DNA haplotypes, one really does not find Amerindians to have one dominant haplotype, like 100% frequency. No such thing exists. Amerindians are mostly haplotype A, B, C, and D. So one do not find the central type with modern haplotypes in America. So we have reanalyzed the modern data, and we found the central type to be haplotype R. So that haplotype actually the most ancestral type of R is R0, and R0 is more common in East Asians. It's not common in Amerindians.
Okay.
So that's for mitochondrial DNA. And then for Y chromosome, you also do not find a central type in America. And the most common type, central type usually generally means it's also the most common type among people on Earth. The most common type is haplotype F. And the root of F is in East Asia. This has been independently concluded by several different groups. We obtained the result, and as well as other groups have no problem. The root, the origin of F is in East Asia. It's not in the Middle East or in Europe, where you would have expected a divergence to be in Middle East if the out of Africa model is true.
Okay. And since 2010, we've had an explosion of ancient DNA studies. And that provides, a legitimate kind of litmus test, to the out of Africa thinking versus the out of Asia thinking. What is your assessment of what we learned from ancient DNA sequences, Tianyuan, Ustashim, and others, that you believe, support the out of Asia theory and weaken or disprove the out of Africa theory?
Yeah, I believe the ancient DNA is a very powerful test of these models, and it has effectively killed the out of Africa theory. Because we would expect the earliest modern humans in Europe and East Asia should be most like Africans, should carry some African genetic similarities, right?
Yes.
And yet they show no evidence of that. And more informatively, the earliest modern humans in Europe actually carry East Asian DNA, so fully consistent with our out of East Asia model.
Yes, yes, I agree. That's exactly my reading. That was a major surprise.
Yeah,
And
yeah.
yeah, and another thing that I noticed is when so the out of Africa, comes with an additional theory of a serial bottleneck coming out of Africa. So the explanation for the limited genetic diversity outside of Africa has always been that there was a bottleneck at the exit from Africa. But when we looked at the ancient DNA diversity from, say, Lojbur, the Lojbur site in Europe, we didn't see high genetic diversity in Lojbur, higher than that of modern Europeans. We saw a lower genetic diversity in Lojbur compared to modern Europeans. We suggested, that the excessive genetic diversity in Africa probably has a different cause, just like you pointed out, a different cause, than just the time from the most recent common ancestor. So it may be admixture, maybe the mixture of different population drove the diversity upwards in Africa, like it did in Europe compared to Lajbur. So it's ancient DNA came up, with some critical findings that I think largely disprove, out of Africa.
Yeah, the bottleneck hypothesis interpretation has traditionally been considered to be very strong evidence for the out of Africa model. But genomic studies in recent years actually do not support the bottleneck hypothesis. First of all, Australian Aboriginals has the highest genetic diversity next to Africans. So this is not expected because the distance of Australia from Africa is very far, and the serial bottleneck hypothesis predicts Australian Aboriginals should have a very low genetic diversity.
Great
And
point.
secondly, we can see similar genetic diversity in the truly neutral variants, and that's not consistent with the bottleneck theory. And finally, different populations share a lot of variants, but the sharing is mostly focused on fast evolving variants. The slowly evolving variants are not shared to a significant extent. And the bottleneck theory cannot explain the difference between the slowly evolving and the fast evolving variants.
Yeah. Okay, Dr. Huang. So my last question is related to another hot topic in modern human origins research, namely that mixture between modern humans and ancient hominins, such as Neanderthals and the newly discovered species of Denisovans. So, what have you found when it comes to that admixture hypothesis? Are you supportive of admixture? Are you agreeing with how many admixtures have happened with the out of Africa thinking, because depending on the author, we encounter either one major admixture event or two major admixture events. So, what are your thoughts here?
Yes, based on ancient DNA studies, there are indeed some evidence for admixture using the popular methods of detecting admixture, even though I disagree with the methods because the methods is based on the neutral theory. So what we find in today's humans, the like 2% Neanderthal ancestry, that could be partly due to admixture, but partly due to natural selection and sharing of variants, maybe just due to convergent evolution. Okay. So the ancient species, the ancient DNA of Neanderthal and Denisovan, they actually provide a good explanation for why in East Asia, in the Oceania, why you have people that look like Africans. And they, by the way, happen to have also the highest amount of Denisovan DNA, like Papuans and Australian Aboriginals. So both in terms of DNA and in terms of traits, phenotypes, the Oceanians similar to Denisovans. So we have suggested in 2017 paper that the ancestor of Oceanians, Australian Aboriginals, Papuans may have been the Denisovan species. And there are more evidence in support of this idea since then. So Denisovan is most likely, we have concluded that Denisovan is African because their DNA are very similar to African. And indeed, a recent paper predicting traits based on genome DNA sequence, they have predicted that the African fossil Cavalli or Broken Hill 300,000 years ago actually is a top candidate for the most Denisovan-like fossil or skull. And this method of prediction has been vindicated or proven correct because they have correctly identified the Harbin skull in China. A Harbin skull in China also is the most Denisovan-like in the predicted traits. And this prediction has now been just proven by a paper several weeks ago in Science that Harbin skull, the sequencing of Harbin skull turns out to be indeed they're genetically Denisovan species. So this implies that the African skull Cavalli is also a Denisovan. So we are the first to suggest that the Denisovans are Africans. Yes. And their descendants ended up in East Asia, Oceania. So the group in Israel predicted traits for the Denisovan, the cranial facial traits of Denisovan based on the genomic DNA of Denisovan. And they have like 14 traits predicted. And then they analyzed the traits of all these known human skulls from 300,000 years ago to 100,000 years ago. And they found like 2 or 3 skull fossils to be most Denisovan-like. The top 2 is, one is Harbin from China, the other is Cavalli from Africa. Mm-hmm. And Harbin has been proven to be Denisovan based on proteomic sequencing and mitochondrial DNA sequencing. And so we can infer the Cavalli prediction is also going to turn out to be correct. And consistently, we have shown Denisovan to have African similarity in DNA, not only in autosomes, but also in mitochondrial DNA and in Y chromosome. The Australian Aboriginals and the Papuans, they have a most common Y haplotype called type C. And type C actually groups with African haplotypes like A, B, and D. So A, B, D are mostly Africans or people who have African traits. And C actually groups with A, B, D, E as part of the mega haplogroup called A, B, C, D, E. Now, A, B, C, D, E shares like 150 variants in common. So in this regard, you can say that C is African-like haplotype.
Okay.
So, yeah.
Okay. Well, Dr. Huang, I think we've covered a wide terrain today and have had the benefit of listening to your detailed presentation of the maximum genetic diversity and the recent out of Asia theories. I certainly believe that American and European evolutionary biologists, anthropologists, geneticists, and scholars of modern human origins broadly should know about all these developments. Monopoly is bad for economy, as we know, though all companies strive for it, and it is bad for science. Diversity of opinions and financial support for theories that might be thought of fringe and that I would personally call startup theories are critical for scientific progress and a healthy public discourse. Thank you, Dr. Huang, for an enlightening conversation. Best of luck to you and your colleagues.
Yes, thank you, Jim, again for providing this opportunity to share our perspectives on evolution and human origins. While we are confident in the robust evidence supporting our theories, we acknowledge that this evidence may not yet convince everyone. As our views gain traction among researchers, we anticipate that new tests will be developed to scrutinize our hypotheses. The accumulation of rigorous testing and additional evidence will be crucial for our theory to achieve widespread acceptance within the scientific community. Thank you again.
Thank you very much. Bye now.
Bye.